Thanks for the nice introduction, and it's my great honor to be here to present my experience on the immunogenicity assessment for the large molecules. Actually, before my presentation, just standing here really remind me when I joined Roche and as the immune safety lead for the Shanghai site, and I tried to find a vendor for the immunogenicity assays to support the local large molecule projects. And I asked around my former colleagues or friends in the biotech or biopharma in China, and actually none of them ever heard MAPS assay or DCT assay. And within few years, now thanks again to the ProImmune, we have the first specialized immunogenicity meeting commencing here in Shanghai and China. I think it's really great progress. And actually, also, as illustrated on the slides now, although I moved away from the Roche job and also to a new modality with, I would say, has lower immunogenicity concerns as siRNA, but I think in this meeting is a great chance to briefly summarize and share my experience as a local user for the immunogenicity assays. Okay, at the beginning, disclaimer, so all the content are based on the public available information and solely based on the personal experience. So speaking of immunogenicity, it's not always bad, as we already heard several times in this meeting. For some modalities like vaccine, actually it's designed to boost the immune response and protect from certain pathogens or cancer mutations. What we cared is the unintended response, like for the large molecule or cell gene therapy. The non-self part can be recognized by the immune system, present by the DC, and interplay with the T cell, and active the T cell. T cell further interplay with the B cell, mature the B cell, and start to express the ADA against the therapeutic protein. And nowadays, we have several tools can assess the immunogenicity, also listed here. I do not need to deep dive into the mechanism because we already heard several times in this meeting. So basically, the first step, we have DC activation, and then we have peptide-based or MAPS assays, and last is whole protein-based assays. Oh, sorry. But the truth is, what we focused or what we have in hand as a tool mainly focus on product-related risks or sequence-based risks. And even on this product-based, we are still lacking of some further tools to assess the B cell or TB interplay. Although team yesterday already briefly discussed some exploratory B cell-based assays, but I think none of them are well established yet as what we used as DCT or MAPS assay. The other factors like patients or treatment related, the genetic background or the immune status, like generally we think the autoimmune disease have the overactive immune response and the oncology patients have the suppressed immune response. Or for the route of administration, for the convenience of the dosing, currently there's a trend we always choose IC over the IV injection, but generally IC is considered as a higher risk for the immunogenicity. So basically for patient and treatment related factors, we are still lacking of proper tools to assess. So I think the question worth of millions is, to what extent can in vitro genicity data be relied upon as a definitive no-go criteria for a non-clinical candidate? I need to, frankly speaking, I still do not have a clear answer to this, but have some thoughts during my following slides. Oh, sorry. Before I answer this question, I want to set a wisdom word from George Box. So all models are wrong, but some are useful. I think as a non-clinical person, we always like to set this sentence because for toxicology study, we also being asked for the predictability from the immunotox study. That's why we usually use two species, one rodent and one non-rodent to do the tox study to get an integrated safety assessment. Actually, it's the same for the immunogenicity assays. We already heard these assays a lot, but each one has their self pros and cons. Like for in silico, it's quick and cost effective, high throughput, and do not require physical material. But generally, although based on the parameter you set, might get different sensitivity, but overall it's oversensitive and lack biological context. And for peptide-based assays, it can be performed at early stage without the final drug substance, but it highly depends on the in silico prediction and limited by the T-cell repertoire of the donor pool. And for the whole protein-based assays like DCT or PBMC assay, it's better mimics the nature antigen processing and presentation of intact molecule, but limited by the HLA and TCR of the donor pool, and requires the drug substance. For MAPS assay, it's the only one who can provide direct mechanistic insight into which peptide being presented, but it highly depends on the HL diversity, and it requires specialized expertise and a high technical challenge. So in an ideal world, we could, from preclinical candidates, we start to do the in silico assay and peptide-based assay, MAPS assay, and whole protein PBMC assay. It's not need to be just one round, maybe several rounds back and forth, and finally we get clinical candidates with minimal immunogenicity concerns. But what I could tell is, from my experience, it's never happened that smoothly. So we always face some pushing back from project team or from the governance meeting. So I listed 3 major pushback or hurdles I faced during my experience. One is the lengthy and costly process. So basically, if we do this all assays sequentially, it will not only invest a lot of money, but from time-wise, it will cost several months. So the first biggest pushback from project team is we need to move this project quick, but we cannot wait for a result sequentially. And also sometimes for the novel formats, the standard methods are developed for the traditional mono antibodies and are frequently encounter some compatibility issue with this assay. And the last but not least is for some non-peptide antigens, which I will also show in my case, e.g. conjugated small ADA against conjugated small molecule. There are actually no suitable in vitro methods simply exist. So for the first hurdle, the lengthy and costly process, if we contact a vendor and ask, we want to do a PBMC or DCT assays, how many donors do you recommend to do this assay? They usually tell you, okay, you need to have 40 or 50 donors to cover all HLA subset. And recently, this European immunogenicity platform published a paper and have a specific section regarding how to select the donor. And actually, in this morning, the keynote speaker also mentioned how should we select the donor. So basically, it's the probability, the 50 donors based on if you do not know their HLA types, if you just randomly choose 50 donors, you should fit the world population HL subset. So when I start to develop the strategy, the first step, we will do it in a 2-step wise. So at early stage, when we have lots of candidates, we will ask CRO to pre-select the HL type donors, which actually is quite easily to get if you buy the PBMC from other vendor, and reduce the donors to 10 to 20, just to exclude the high immunogenic candidates, and can significantly reduce the turnaround and cost. Which on my experience, if we do with 50 donors, again, it's months works, but if we go with 10, we can reduce it to the weeks. And later, once we have some remaining final candidates, we can add extra 40 to 30 donors to fully de-risk the false negative probability. And at the end, I would like back to this sentence, which we cited a lot, and show the full sentence here. So I think my final conclusion is, I do hope the vendor, the sponsor, and the regulatory can work together to move further develop the new methods to increase the predictability for the immunogenicity assessment. And with all of that, I will stop here and happy to take any question.
Thank you, Dr. Yang. Thank you for present some of the clinical side of the cases. And I actually have a rather more general question about also for all of our immunologists presented these 2 days. My consideration is a lot of antibody drugs or peptides now applied subcutaneously administration. And as immunologists, we understand that the immune microenvironment are quite different, like from the IV route of administration versus the subcutaneous administration. So our current assay that I have heard, basically based on a assay that use the PBMC or whole blood or PBMC derivatives in the assays to assess the immunogenicity risk. But considering the subcutaneous injection method, should we, on aspect of the different microenvironment, like you have more Langerhans cells, you have more professional dermal dendritic cells, and your antigen presentation happens in the local lymph node, not in the spleen. So consider all this, is there any suggestion for a more appropriate assay for assess these subcutaneous administration drug of these drugs immunogenicity risk?
So to answer your last question, there are some models which I am aware of. They have this ex vivo skin excipient and co-cultured with immune cells exist, but also to my knowledge, I think it's never used for the immunogenicity assessment. And back to the IC versus IV. I mean, generally we think IC is more immunogenic than IV, but I could also share some paper later. So it's still debate whether IC is really more immunogenic than the IV.
I want to get the guide from our regulatory agents specialist. That do you have any consideration on do you have recommendation or any guideline on subcutaneously administration? Does that route of application impact your evaluation of the data from the industry?
Hi, so Daniela's left, so I'll try and stand in. So the 2014 guidance from FDA does raise the routes of administration as having different potential impacts. I think the interesting thing is when you actually look at products that are given both IV and subcutaneously, there's not a lot of difference. There was a publication, I think in 2024, where a group at Regeneron took all the publicly available data, and I think they looked at 20-something products, and maybe only 2 or 3 of them showed a difference. So I think there's like nice, compelling reasons why there should be a difference, yet the reality is that often there's not. So it can be quite challenging, say, going from the systems we're using, which are maybe practical and at least widely used, to then more complex systems that may not necessarily help. So I take the point that there are cases where route of administration can make a difference, but it would be the kind of thing that you'd, if you were, my take on what you would need to show the regulators is that if you think there's an issue, that you've at least looked into that. So the other piece of data that people often use is the animal studies, and maybe Juhao, you can comment on this as well. That quite often in the GLP tox studies, we're testing very high doses for IV, and then the kind of comparable low dose is the only one that happens in subcutaneous, typically in those GLP tox studies. Often the very high doses IV, there's just too much drug to measure ADA. So it then looks like, across your maybe 3 groups of animals that have received IV, 1 or 2 groups may not show any ADA. basically you can't measure them. And then you get to the lower dose and they're pretty much equivalent. So we did do a study of Roche data to look at this question you were asking in the NHPs. And what we found was that basically with the molecules of interest, everything was really high. And so we did get equivalent high immunogenicity from both IV and subcutaneous. Do you want to add anything, Juhao, to that?
I think you perfect answer, yeah.
Okay. So we can follow up on any detail as well. Thank you.